Let's continue here with the DNA replication. So this slide here was about the base pairing rules between A and T and G and C. So remember, the goal in replication is to make two strands of DNA where there was only one, and it's always done in preparation for cell division. So we need to copy the entire genome before a cell can divide. Okay, so in DNA replication, one strand serves as a template for the other. For the second strand, there are a whole host of enzymes that you need. The most important one, of course, being DNA polymerase, but you also need accessory enzymes, and there are some listed here: topoisomerase and gyrase. They basically help the DNA to unwind properly, and then helicase will separate the strands, the double strands, from one another, and then you need to create a replication fork. So this picture, it's not super obvious how this is a fork, but kind of picture it this way. You have a double strand of DNA, so like this, and then you start opening it up like that, creating what's called a replication bubble. And let's say you were to cut this bubble open, then you would have more something like a fork that looks like this, something like that. Okay, we'll see other pictures where it's a little bit better. So anyway, this is a replication fork where we're opening up the double strand, and then as soon as the double strand of DNA is relaxed and opened up, you can add, from an origin of replication, you can add nucleotides, new nucleotides, using DNA polymerase, and then filling in the secondary strand with new nucleotides. So here's our enzyme that's doing most of the job. It's called DNA polymerase. It adds nucleotides always in the 5' to 3' direction, and it's kind of a problem that it can't go the other way, but you will see. So it's always in the 5' to 3' direction. You need to initiate the formation or the synthesis of the secondary strand with an RNA primer. Typically, we start at an origin of replication, and in the leading strand, it's not a problem. You will see in the leading strand, the DNA is synthesized continuously, starting from this origin of replication, then all the way just keeps going. And the lagging strand, we need to go backwards, basically, because DNA polymerase can only synthesize in 5' to 3' direction, and you will see how that is going to be a problem. So we need to synthesize the lagging strand in pieces. It's called discontinuously. You need to first start always with a priming piece, and that's an Okazaki fragment. That's a little RNA primer. And then you need to fill in the nucleotides using DNA polymerase, and then cut out the RNA primers that you used to start it out, and then replace that with DNA nucleotides. Anyway, so here is a list of enzymes that are important for DNA replication. As we go through the process, you will see which ones are important for you to know. So the DNA gyrase together with the topoisomerase down here, these guys help to relax and unwind DNA. The main enzyme, of course, is DNA polymerase, and we will also talk about some of the other enzymes as we go through the process. Okay, so here this one looks more like a replication fork now. And basically what happens is that you're starting out with this double strand piece of DNA, and then you have an origin of replication, and basically, let me just kind of draw a diagram of this up here. So you're starting out with a piece of DNA, so I'm just gonna make it just one piece right here, so double strand, and now we want to copy this. So we're going to first open up the strand. So we have an origin of replication, let's say right here, and we're opening up the double strand right there. So then what ends up happening is, looks like this. It creates this bubble. Now what you have here is basically what I drew up here. That would be the extension of this thing here, where you can see where the DNA polymerase is making the secondary strand. It's just start from a starting point. Let's say that starting point was up here. On the leading strand, we're going to start, I'm using a different color now. We're going with DNA polymerase all the way, and then we need to open up more and more pieces, and you can see how this is pretty easy. Everything's very straightforward here. On the leading strand, that will be this part right here. So you started right here, and it's already done right here. The DNA, the new half old, half new DNA strand is already rewinding and looking all like a ribbon. Here it's still straightened out. That was the work of the helicase, unwinding a stretch of DNA. And here's your DNA polymerase, the main enzyme that's just zipping along here and creating a secondary strand using the base pairing rules. So that's all easy. That's the leading strand. And here on my bubble, on this down here, the leading strand on the other side will be going in this direction. Remember, DNA polymerase can only go in one direction, so we're going in this direction. So nice and dandy right here, all easy on the leading strand. Now the lagging strand, not so much. The problem here is that you can still only go in one direction. So if, let's say here on this part where we started here on this origin of replication, you can't just go in this direction because the DNA polymerase can only go in 5' to 3' direction. And same on the bottom here, you can't just go in the opposite direction. So the only way for you to finish this part right here and this part right here is to, well, use Okazaki fragments. So in the diagram right here, it's not super clear. I feel like we need to put down this RNA primer. That's this piece right here with the primase, and then we need to use DNA polymerase to fill in the pieces. On my diagram, the way it would look like is as follows. If I want to finish this secondary strand right here on this piece of DNA, then I need to first put down an RNA primer right here, and now I can come in with my DNA polymerase and finish this piece. Then I have to come back and replace that primer that was RNA. I need to replace it with DNA, but you hopefully get the idea. So, and in order to finish this up, we need to set down another RNA primer and then fill in this piece for the secondary strand. Up here, it would be the same thing. You can only go in this direction, remember? So you would have to put down a primer right here, and then you can start filling in the gaps right here. And here would be another primer, and then you fill in the gaps. So that's the lagging strand. And of course, it takes more time because there's more things to do, and therefore we'll call it the lagging strand because it just takes longer. Okay. The energy for the DNA replication is supplied by nucleotides, mostly via ATP. So you need to hydrolyze a lot of ATP to power this the replication process. And here will be the finished product. So you have the template strand, and then you're making the new newly formed strand. And of course, it does cost you some energy. So then at the end, we're going to have two brand new DNA molecules. In bacteria, it's relatively easy actually because you're only dealing with one circle. We're going in both directions, and once the two halves meeting up, basically that's when you're done and you have a complete replication of the entire genome. DNA polymerase is amazing. It is an enzyme that doesn't make hardly any mistakes because there's a proofreading function as part of that enzyme. It doubles and triple checks everything it does, and so the DNA replication is with high fidelity. It's a DNA polymerase makes hardly any mistakes. Now, of course, any mistakes that it would make, that would be considered a mutation. And here you have this whole process of replication summarized. Up here, it's a electron micrograph where you can see the DNA stained right here, the two new stretches, two new circles of DNA. Here in a diagram, it's not super clear, but you have this origin of replication. It's where we start out right here, and then you start the replication here, make this replication bubble a fork, and then you just continue in both directions until you meet up down here. That's when it looks like this, and then you just have to finish up the coming back to the full circle here, finish up and separate the DNA into two separate molecules, and then the cell is ready for cell division. Okay, so that's replication. Now let's move on to, we're going to move on to protein synthesis. And so in protein synthesis, we need to make messenger RNA first in transcription and then translate that into proteins via translation. The transcription part first, for that we need to make messenger RNA, and RNA is slightly different. Here's a summary of the difference between RNA and DNA. In RNA, RNA is typically single-stranded, and it uses the 5-carbon sugar ribose instead of deoxyribose that we have in DNA, and it uses uracil, the base uracil, instead of thymine. Now for transcription translation, we need actually different types of RNAs. The one that everybody knows, probably intuitive, that's the messenger RNA molecule. That one carries the information, so that's the copy that carries the information from the DNA to the ribosome for translation. Now, for translation, you need transfer RNA, and we will see that. And of course, we need ribosomal RNA. So the ribosome itself is composed of ribosomal RNA. So a ribosome is ribosomal RNA. Okay, now let's take a look at the process of transcription in prokaryotes. It's actually so much simpler than in eukaryotes, so you're lucky that we're doing prokaryotes. And the initial knowledge was acquired using prokaryotes anyway, because they're so much simpler. So the transcription, basically we're going from DNA to messenger RNA, and the process is known as transcription. We need to start at a regulatory sequence. It's called a promoter. That is basically sort of like the switch for turning something on or off. And starting with the promoter, we're assembling a bunch of proteins as enzymes that then will make us a messenger RNA transcript. Now, transcription proceeds in 5' to 3' direction, just like we had in replication, except that we're using a different enzyme. We don't make DNA, so we don't need DNA polymerase, but we need to make RNA, so we need RNA polymerase. And then, so transcription is initiated at a promoter sequence, and then it will end at a terminator sequence. Okay, so let's take a look at this. Here will be a stretch of DNA that you see right there. The goal will be to make a transcript, a copy of a gene that then can be translated into protein. So we're starting out here with our DNA double strand. It's kind of tricky to know which of the two strands is actually the coding strand, and we don't have time to get into that, but something for you maybe to think about. If you can't sleep at night, think about how a cell might work this out and figure out which one is the coding and which one is not the coding strand. So anyway, let's keep it nice and simple. Let's assume that we have figured it out, and this lower strand right here is the coding strand. And so now we need to make a copy, an RNA copy of that strand. And then we're using the enzyme RNA polymerase once we have found our promoter sequence. So here is our promoter sequence, and the gene begins right here. That's called the transcription start site, the TSS, transcription start site. And we're using the enzyme RNA polymerase and making a RNA copy of the coding strand until we hit the terminator sequence. And that's when the RNA is complete, and it will then have to be processed, and then it can be translated. The information gets translated into protein. So translation is where we take that messenger RNA molecule and translate it into the language of protein. So the term translation is really, it depicts it pretty good, because we're going from the nucleotide-based language of DNA and RNA, we're going to a different language of amino acids that makes then proteins. A couple more terms that you need to know about this. Codon is a group of 3 messenger RNA nucleotides, a set. It's like a code word. Think of it as a 3-letter word that specifies a particular amino acid. Well, if you think about how many code words can you make, if you have a 4-letter alphabet, so you have A, G, C, U, you want to make 3-letter words, so you have 4 times 4 times 4. That would be 64 possibilities of making 3-letter words with a 4-letter alphabet. And let's take a look what that looks like. So we have 61 sense codons and 20 amino acids, and that will be the genetic code right here. And we'll pick it up from there in the next